Okay. Who's ready? So hopefully you all got your grades online. I'll have the exams in recitation. And for those of you not in my recitation, feel free to come visit me, either at the beginning or end of one of my recitations, or in my office whenever. Yeah. So people did fairly well. Oh, I meant to put up the distribution. But actually I should stop doing that because it might make people feel bad. We don't want people to feel bad. But the average was an 81, so that's good. Okay, so there was a change I made to the quiz this weekend because basically to limit sharing of correct answers among students, there's a way to make it so you can't see the correct answers until the quiz is over. You can go in now, I believe, through Grade Center and see what you got right and what you got wrong and blah, blah, blah. But that way at least someone can't go, screenshot, here you go, guys.
I can still see. I was trying today.
Has anyone been able to see? You saw it, right? Yeah, so people have been able to see it. So maybe after class you can catch up with Marika.
Yep.
Over here and tell her how to see it.
So you can see it like after Monday.
So yeah, so basically after it's due on Monday, you can go in and see it. And if you have trouble, you can come to my office so we can work through it together.
You just go through the grade center.
Yeah, go through Grade Center.
Just click on your score.
Okay. Apparently you click on your score and then you can see it. So sorry for the confusion. I meant to email everyone about that, but my daughter was puking, so. So it's just like, oh, quiz. Parenting. Okay. I think there's something else I was going to say. Oh, so someone said something about weather. On Thursday, there's not going to, I don't think there'll be any weather that we have to worry about on Thursday. So don't worry about it. You guys won't miss recitation. I just checked right before I came in here, and Dan Burke says, but I checked them all. They all say it's going to be south of us, so we should be fine. Who's ready for spring? You know that daylight savings is this weekend.
Is it really?
Yeah. Shocking. Shocking. It just doesn't feel, it feels like January, so it's shocking.
Exactly.
Yeah, so things to look forward to.
Not what I needed.
Okay, that's generated a lot of commenting. Okay. Okay, so far, sorry, I'm wasting time here. So far, this is what we've talked about. Just introducing the concepts in cell cycle, the different phases, checkpoints. I talked about different methods for study, BRDU, PI staining, which is the flow cytometry, conditionals, temperature-sensitive mutants, visualization, just looking at the cells, and immunofluorescence. I spoke about different ways of regulating CDK activity. Remember, CDKs are what's going to regulate which phase of the cell cycle you're in. Cyclins, WEE1, CAK, cyclin activating kinase, and CKIs, cyclin kinase inhibitors. And then we spoke a little bit about the ubiquitin proteasome system, about ubiquitin ligases, and the cyclical proteolysis of proteins to regulate the cell cycle. And that is where we stopped. Yeah. Yeah, the question about ubiquitin, yes.
Yeah. So, you had a slide up last week, I'm sorry, visual, and it was like E1 ubiquitinates E2, and then E3 somehow ubiquitinates the substrate. Is E3 being ubiquitinated as well, or is it just transferred?
Yes. So the question is about the enzymatic reaction behind ubiquitination. There's E1, E2, E3. E1 activates ubiquitin. Then it gets transferred to E2. It's not being ubiquitinated. And then it gets transferred to E3 or binds to E3. Some E3 actually bind to the E2 ground ubiquitin. And then it gets transferred to the substrate, and that's the ubiquitination reaction. That's the covalent modification of the substrate. So E1, E2, E3 is kind of just this handing off process and activation and all that. But that true ubiquitination that results in the post-translation and modification of the protein is done by these three. Okay? Yes. Any other questions before we move on? Things that you were thinking about? Some people have started watching my lectures from last year. I highly recommend that if you want to get kind of a head start before taking the quiz. It's not a bad way to do it. They're all on YouTube. Okay. So, cyclin CDKs actually regulate the proteasomal degradation of specific substrates. So I spoke about ubiquitin ligases and how certain things are degraded to turn off processes. The biggest example in cell cycle would be cyclins being degraded cyclically. But also, other proteins are degraded to regulate the cell cycle other than cyclins. And one method of regulation of that degradation is by phosphorylating the substrate. So one of the classes of ubiquitin ligases that we spoke about, the SCF, skip-cullen F-box ubiquitin ligases, they recognize ubiquitinated substrates. You always have to wonder, well, how can you regulate whether or not the proteins can be recognized and ubiquitinated, right? So with SCF proteins, so here's your substrate, your target protein in the middle, and what happens is it's phosphorylated by SCDK, and then that phosphorylated substrate is recognized by the SCF ubiquitin ligase, and it's polyubiquitinated and degraded. We'll get back to that in a minute. Okay. So another ubiquitin ligase that I introduced, APC. So there's two main ubiquitin ligases that are involved in regulating cell cycle. SCF recognizes ubiquitinated substrates, and APC, which is regulated by CDC20, right? So APC, anaphase promoting complex, it's required, its activation is required for progression from metaphase to anaphase so you can exit from mitosis. So that specific ubiquitin ligase degrades certain things that are required to be degraded so that you can exit mitosis. So here's an example where you would be, this is kind of an example how you can use these different kind of mutants. Were you raising your hand?
Yeah, you said the two main types were FCS and APC. What is APC regulated?
APC, I think, do I have it coming up again? I do. But it's regulated by another protein called CDC20 that binds to it and activates it. So normally APC is present but off. And when APC is present but off, but CDC20 will come and bind to it and activate it. And then APC can polyubiquinate all of its substrates. So this is how you might use the whole conditional mutants, or can we see? Okay. Sometimes these things get very high. You might use a conditional mutant or a temperature-sensitive mutant to study the conformation or the morphology of yeast under different circumstances. So here is an example. You can have wild-type cells, and then you would have CDC16 mutant cells, so they don't have an active APC. And so if you look at the different cells, what would you, and so this could be, if it's a conditional mutant, let's say it's a temperature-sensitive mutant, at your permissive temperature, it would look like this. And at your restrictive temperature, it might look like this. So what do you guys see if you compare the different cells?
It's not meaning spindle fiber correctly to move electively in the DNA.
Okay, so it's just a picture, so you're not going to see the mitotic spindle itself, but I think you're saying there's something wrong with mitosis. Okay?
It's stuck at one phase.
They're all at one phase. I think I heard that over here too. They're stuck at one phase. Good. What phase of the cell cycle is it?
Basically stuck in it.
Yeah.
They can't progress to anaphase,
Right. So they're having trouble going to anaphase, but you can't really see that because you don't see the chromosomes. What do you see? The DNA is not splitting. right? So you see nuclei only in one cell of the two that are stuck together. Yeah.
Maybe it would be, I don't know, it's right now. Maybe it would be saying something.
Right, so you're not seeing actual true budding, because this is supposed to be budding yeast, which is why you can see these little buds coming off. But yeah, it's not finishing the budding process. You can say there's an issue with cytokinesis. Okay. What else? I think that was pretty good. Well, yeah, so exactly, these cells are defective for APC, so they're not degrading S-cyclin, they're not degrading M-cyclin. N-cyclin degradation is required for exit from mitosis, so they're not exiting mitosis. Yes, you're not really saying, you can say you're not seeing chromosome segregation because you see nuclei kind of just in one of the two. They've all gotten to the same phase and stopped, so they're arrested. And so one of the targets of APC is securin. So normally you've got this inactive APC, and CDC20 will bind to and activate it, and then it goes and degrades all of its substrates. And one of its substrates is securin. Securin is an inhibitor of an enzyme called separase. And what separase does is it goes in and it cleaves cohesins. Cohesins are proteins that keep sister chromatids together. Does it make sense to cleave your cohesins prior to anaphase? Do you want to cleave your cohesins prior to anaphase? Who's saying no? Why no?
We don't want them be, until after anaphase, we don't want them be separated, we want them line up.
Well, so anaphase, what happens to your chromosomes during anaphase? They separate. So you need to cleave your cohesins prior, otherwise you can't separate your sister chromatids. So that's what's happening. Normally in G2 and in metaphase, you've got your sister chromatids held together and they line up, and the mitotic spindle binds to kinetochores. And then your separase, so between metaphase and anaphase, separase comes in and cleaves all those cohesins so sister chromatids can separate.
So is this one of the examples where you said that ubiquitination could be activating as well, because it's activating essentially
Yeah, well, so the comment is this an example of how I said ubiquitination can be activating and inactivating? Sure, I wasn't thinking of it that way, but yeah, so ubiquitination is degrading something. Sometimes it'll degrade something to turn something off, and sometimes it'll degrade an inhibitor to turn something on. And here what it's doing is it's degrading securin, which is an inhibitor, which turns on separase, which cleaves cohesins, which allows for progression to anaphase. There are also other things that are degraded. Yeah.
So CDC20 moves APC out of the way.
wait, what?
CDC20 NFH1 complex.
Mm-hmm. Yep.
And once you degrade it, you can move forward.
CDC20 activates APC. What you degrade, you don't degrade CDC20. What you degrade is securin. Yeah. So once you degrade securin, separase is active to move forward. It also degrades M-cyclin, sorry. So you can think that actually activation of APC creates this negative feedback loop because MCDK activates it, and then MCDK is turned off through degradation of M-cyclin. So that's APC, yes.
So I'm just curious, what's that extra C for?
Oh, yeah, sorry. So depending on the book you look at, I tend to call it APC, but sometimes people call it APCC. In the textbook you'll see it sometimes. It's basically, I'm not sure, I think it may be, I don't know why. It's anaphase promoting complex. And so I think I was trying to stay consistent with the nomenclature the book uses. But I'll often refer to it as APC because it sounds weird to call it APC. Yep.
So this is an example that doesn't apply to all CDK regulation, but it's like one example of a certain type of CDK. Right.
Is APC, activation of APC an example of one kind of
Like,
cyclin CDK regulation?
how general is this? I guess this is a really specific example of one kind of CDK.
So this is an example of one, so if you want to generalize, what your way of generalizing would be to say polyubiquitination of substrates are involved in regulation of cell cycle. Cyclins are degraded by ubiquitination, which is carried out through either SCF or APC. Because actually, so APC, so the question is how much can we generalize this, and how is this just a specific example? And so APC, and I don't get into great detail because it's not cell biology, but half the cell cycle APC is active, and the other half pretty much SCF is active. And so one of the two is usually degrading a cyclin or some other. There are millions. Actually, I have a list of substrates that's going to come up. Not millions, but there are many, many substrates. And so what I'm telling you here is APC, when it's active, degrades M cyclin, it degrades S cyclin, and it degrades securin. And those are all things that are required for moving forward in the cell cycle in different ways. Make sense? And as you will see shortly, it's also involved with S phase, and then SCF is also involved with activation of S phase. Any other questions? Okay, so M cyclin activates CDK. M cyclin CDK activates APC, and then targets itself for degradation. So actually, the M cyclin, the phosphorylation activity by that MCDK is involved with activating APC. But then the APC, once it's active, actually goes on to polyubiquitinate M cyclin. And that promotes exit from mitosis.
You said M cyclin CDK activates APC, so how does it do that? By
It
activating the
phosphorylates, I believe it phosphorylates CDC20, which encourages interaction with APC, but don't quote me on that.
Okay, I'll double check it.
Yeah.
Sorry, I must have missed it. Did you explain the negative feedback?
Negative feedback because M cyclin activates APC. M cyclin CDK activates APC, but then APC degrades M cyclin, which turns off MCDK. So that's negative feedback. Yes. Yeah, negative feedback. So that's when something turns itself off, right? So M cyclin CDK turns on APC, but APC degrades M cyclin, so it turns itself off. Make sense? Good? Okay. Okay, so we'll leave that for a second. Okay, so we talked about checkpoints earlier on. I'll just bring them up again once more. And then we probably won't touch on checkpoints again until we start talking about DNA damage and repair at the end of the semester. So, but it's just important to acknowledge that when things go wrong, the cell cycle is halted by activating checkpoints. Okay? So you got your spindle checkpoint, you've got your G2M checkpoint, S phase checkpoint, start checkpoint. These are all important points where the cell is checking itself to make sure things are right. There's no DNA damage, there's no stalled replication fork, so you don't want to progress to the next phase unless things are right. And if things are not right, then halt the cell cycle. Okay? It's the basic idea. Okay, so this brings us to a quick practice problem where I'm making you guys look at the morphology of cells. I'm making you guys think about mutants and checkpoints. Okay, so this is the response to DNA damage, and we'll probably come back to this again at the end of the semester. And so when they were trying to identify different genes associated with checkpoints, they were looking at these genes that are called RAD genes that are radiation resistant genes. And what they did is they've got wild type cells, so these two are wild type. And then this RAD9 mutant, and that's these two down here. And up here it says before irradiation, so this is before they were irradiated. And then here's 10 hours after irradiation here. And so what they did is they took the wild type and the RAD mutant cells, and they grew them normally, and they plated them, and they took a picture, and then they irradiated them, and they let them grow, and then they took another picture. Irradiation causes double-strand breaks in DNA, so it's a DNA damage agent, I guess, function. So the question is, if you compare all of these, compare this to this, and then this to this, and these two mostly, what does this tell us about the role of RAD9 in the response to DNA damage and activation of checkpoints? So you need to think about how checkpoints are there to check the DNA, make sure all is well, in order to progress your cell cycle. And look closely at post-irradiation for both wild type and mutant, and try to see if you see any differences. So go ahead and chat in small groups and try to figure it out, and then I will come around and help. Or we'll go first. So imagine our actions so it's the same cell, so they're all so you can see here's a single cell, what happens here, compared to a single cell, what happens here. It's not like a significant amount,
Well, actually, I don't have.
And it's professional, I feel like they're dumb.
It's gonna be raising the hard one.
And then it's me, and then it's you, so that kind of makes sense. That's what I'm saying. She just.
Yeah, I thought she was like going like. That's all I have in my life, you know what I mean?
So we've been or if
Then you tell us, yeah, like, all then you promote apoptosis and you can't fix the problem.
That's what I was thinking.
Okay, how's everyone doing? Does anyone have any questions? I have lots of questions about how to look at this, whether or not, like, what you're really looking at. And so something to take note about is, for example, so here you've got one cell, right? And it becomes two cells. That's with wild type, two huge fat cells. And here's one cell, and it becomes 6 normal sized cells, right? And so you have to ask yourself, what might Rad9 be doing? So, who has an idea? Yeah.
Well, it seems more like the Rad9 is involved in recognizing the damaged DNA and then halting the cell cycle when it recognizes it. And so when it's mutated, it either doesn't recognize it or it lets it happen anyway. So it ends up with more.
Okay. Okay, so I'm hearing Rad9 must be involved in recognizing in some way DNA damage and activating a checkpoint to halt cell cycle, because when it's mutated you see replication anyways, and in wild type you don't. And so that's what you said. In the presence of DNA damage, you're still seeing replication when Rad9 is damaged. So this one is a little bit challenging because you have to think backwards, right? Because the wild type should halt DNA damage, should halt cell cycle in response to DNA damage, and that's what you see when you look at one becoming these 2 big fat cells. There's like something, they got to a certain point and it couldn't proceed any further. And you can't, and if I just had this by itself, you wouldn't be able to tell me, or just had Rad9 by itself, you wouldn't be able to tell me much. It's only when comparing to the control wild type that you can really tell anything. And so here, so when Rad9 is gone, that checkpoint is gone. So you can bypass the checkpoint. And so even in the presence of DNA damage, you're getting replication.
I'm confused. So you think of it in the Rad9, it's gone because of the DNA damage?
Rad9, so the Rad9 cells are Rad9 deletion mutants. So they do not contain Rad9. They are missing Rad9.
So now can you tell us what Rad9 is?
Well, you're supposed to tell me what Rad9 is. I'll get to that in a minute. Yeah, yes.
Specifically, could you say that Rad9 would be involved in, like, it's messing with the APC somehow? Because if it goes all the way through, so that means,
So it's hard to know exactly. Okay, so the comment, guys, let's try to listen to each other for a minute. So the comment I just got down here is that, could you say that it's messing with APC because when Rad9's gone, it's going all the way through where it's stopped. You're not seeing exit from mitosis here, right? You can't say that for sure. You could say something is preventing exit from mitosis, or something is preventing progressing through cell cycle, but you would have to probably do some more experiments to figure out which thing. I saw more hands up.
So does the wild type have?
Yes, wild type has Rad9. Rad9 mutant has no Rad9.
Okay, so then without the Rad9, it just bypasses the checkpoint, and that's why it's able to.
Yep, without the Rad9, it bypasses the checkpoint and is able to replicate. The purpose of a checkpoint is to stop replication when you have DNA damage, and we've irradiated these cells, and so we've created double-strand breaks. You should get halted cell cycle. Yes.
But what about for someone like in the wild type, like in the center where there's like a huge cluster of cells?
Like this?
That one. How can you
So the most, okay, so you're saying you're having a hard time reconciling cell cycle arrest with a big cluster of cells, right? So what you need to do is you need to do the comparison between wild type and mutant. Because I also, the first time I saw this, I was like, wait, I'm going from not so much to a whole lot. How are you telling me cell cycles arresting? But the difference is, so this one had 1, 2, 3, 4, 5. And I don't know, I guess we can count these. But so here you're starting with 5. And so you're getting a small cluster. But if you compare 1, it's easier. So 1, you get 2, whereas 1 here you get 6. And then the other thing is I guess to see an equal effect on everything, you would have to start with single cells everywhere, and then make sure that each cell is equally exposed to have the equal amount of DNA damage, and so it's not actually perfect. A perfect way. So if I drew this, bless you, if I drew this out for you, what I would do is I would have 1 to 2 only everywhere, and then 1 to clusters everywhere for the other one. But this is more how science works, where you're counting and you're looking at the ratios. Yep.
Could there be any possibility that the cells that don't have the Rad9 are able to repair the
So is there any possibility that the cells that don't have mutant Rad9, so these wild type ones, some of them are able to repair damage and move on? Yes.
Could that be why?
Yes, so depending on whether or not, these are yeast, and they have, depending on where they are, their cell cycle, sometimes they're diploid and sometimes they're haploid, and if you're haploid, you have no way to repair, because there's no other DNA to copy off of. So yes, that is possible that some are able to repair. Yep. And that's how you have to think when you're a scientist, is okay, I see this data, what does it mean? Why is it not how I expected it to be? Yes. Okay, Venus, are you happy with the function of Rad9? It's somehow involved with activating a checkpoint, because in the absence of it, you don't activate your checkpoint in the presence of DNA damage. Okay? Okay. Are we happy? Should we move on? Okay, so when you're activating a checkpoint pathway, it requires the activity of 3 different classes of proteins. And they're all logical, right? Sensors, right? So something needs to sense the damage, if it's DNA damage, for example. Transducers, so that's some kind of protein that's going to tell other proteins that there's damage, usually a kinase or a protease or something. And then you have your effector proteins. These are proteins that are going to do something in response to the damage that's been sensed. Those would be repair proteins, proteins involved in synthesis of DNTPs, because repair needs DNTPs, right? Those are your bases, right? And proteins that induce cell cycle arrest. That's just a very, very general way to think about it, but it's a right way to think about it, right? Sense the damage, tell someone there's damage, and let that someone respond in a way that's conducive to repairing the damage or killing the cell, depending. Right, so for example, double-strand breaks, which is what was induced by the irradiation. Single-stranded DNA accumulation, you should never have lots and lots of single-strand DNA for a long period of time. Otherwise, something's wrong. right? Because normally you only have, bless you, everyone's decent today. Often you'll have some single-strand DNA, but that's quickly replaced by double-stranded DNA when replication's functioning normally, right? If you have single-stranded DNA hanging out for a while, you probably have a stalled replication fork. And so that's another sensor. So these are things that are sensed by the cell. I know. I think I'm gonna have to hand out pop drops. You guys better not have any sex. Okay. I'm like ironclad because I've got 3 young children. And so your effector proteins usually are going to do things like prevent cyclin CDK activation, prevent CDC25 activation, things that are going to prevent cell cycle progression. Okay? Did I see a hand going up in the back? No? Yep.
What's the SSDNA?
Oh, sorry, SS, single-stranded DNA. Yeah, I tend to write things in ways like I just think everyone understands it, so always stop me. SSDNA, single-strand. dsDNA would be double-strand DNA. Yep. Any other questions? Yes.
So BRCA1 and BRCA2 sensor.
BRCA1 and BRCA2, are they transducers? Do they activate p53? Okay, you know, I think that I can't remember exactly that pathway right now. So we're not talking about BRCA1 and BRCA2 and p53 and DNA damage repair. And I can't remember exactly right now. But if we go over the pathway, we can kind of pick a plan of all that. If you want to visit me after, or whoever wants to visit me after, we can do that. I just don't want the rest of the class to freak out because they don't know what BRCA1 and BRCA2 are. Who freaks out whenever they hear someone talk about something I don't know about? Yeah. I think it's anxiety provoking. Yes.
When it says stalled replication fork, does that mean that it recognizes it right before it goes into a replication fork?
No, so a stalled replication fork is like, so you've got your bubble, right? So these are your replication forks, right? All right, can you guys on that end see anything? Over there? Yep. So these are your replication forks, right? And so you're gonna have DNA replication going that way. And so when it stalls, let's say there's DNA damage, let's say there's some kind of bulky, some kind of like lesion on the DNA from some environmental insults, from UV irradiation, any of these kind of things that we'll learn about at the end of the semester, that'll cause like either your bases to come fused together that are next to each other, or your some kind of intercalating agent, something that's going to prevent opening up of the DNA and progression of DNA replication will result in a stalled replication fork. And often will also result in an excess of single-strand DNA sitting around. And so those are the kinds of things that are recognized. This is kind of just a taste of this right now, because we're going to go in detail later. But I think it's important to talk about it when we're talking about cell cycle. Make sense? Okay. Okay. So checkpoint activation will activate repair pathways or induce apoptosis if repair is not an option. And we'll talk about this later in the semester. Okay, so let's talk about S phase, which is also known as DNA replication. You guys know that. Okay, so this is the gist of it, right? So you have your origins. Remember, there are many origins, right? We all learned that in recitation last week. You have your origins, and they're primed for DNA replication as early as M phase, believe it or not, or G1. And so what that means is that these CDC6 and the origin recognition complex, CDT1, and your helicase, that pre-replicative complex, which is basically about loading your helicase on your DNA at your origins, that all sets up long before S phase. And it just sits there. And it's not until SCDK gets activated that you get activation of your origin. So your origins start firing then. And what happens then is you get disassembly of these different these helicase loaders and all that. So your CDC6, which is involved in loading your helicase, will be phosphorylated and degraded by SCF. And your CDT1 will be inhibited by a protein called geminin. And your SCDK will also phosphorylate your origin recognition complex. And then that'll help with recruitment of polymerase and all the different proteins required for DNA replication to happen. And then you get DNA replication. So you can see anytime you see phosphorylation, that's being done by SCDK. Remember, you've got your SCDK, that's regulated by cyclin binding. And then once that's on through CAK or CDC25, whatever, or degradation of CKIs, whatever needs to happen for it to be activated, that needs to happen. Then you have your SCDK active, and it can phosphorylate things in order to trigger DNA replication beginning.
You said that was, did you say that was degraded by SCF?
Phosphorylated CDC6.
So SCF can degrade recognition or phosphorylation?
Okay, so thank you. So I just said, Venus said to me, wait a minute. You just said phosphorylated CDC6 is degraded by SCF. I thought only things that are ubiquitinated are degraded. And so if you remember in the beginning of the lecture, I showed you this, and I said SCF recognizes phosphorylated proteins and polyubiquitinates them. So the activity of SCF is regulated by the substrate being phosphorylated and then being recognized by the ubiquitin ligase. Yes?
SCF is the entire complex.
Yeah, skip. So SCF stands for skip, colon, F-box. So it's a multi-protein complex that functions as a ubiquitin ligase. So it's going to recognize a phosphorylated substrate through interaction with the F-box protein, and then polyubiquitinate and degrade it. Yes?
So all of the proteins that ubiquitin ligase interacts with, all of them are phosphorylated. They can't be.
Only with FCF. Only with FCF. Yeah, so that's how you regulate. The question wait, so all the proteins that the ubiquitin ligase interacts with are phosphorylated, and I said only with SCF, and that's because SCF recognizes ubiquitinated substrates. Specific ubiquitinated substrates. Not to say that every ubiquitinated protein is going to be bound and degraded by SCF. There are other factors that come into play. But one way to regulate the stability of the protein, so the one that we just talked about, let's go back over here, is this CDC6. And so I'm telling you it gets phosphorylated by SCDK, and then it's recognized by SCF and degraded. And so that's a way to regulate the stability of CDC6, right? So when SCDK is inactive in G1, CDC6 is hanging out and helping to load helicase onto the DNA. But once SCDK is active in S phase, then it will phosphorylate CDC6 along with other things, right? Also phosphorylates ORC. And then it will disassemble that pre-replicative complex, the pre-RC. Good? Okay.
So I have one more question
Yeah.
about, okay, so you're saying that the substrate is phosphorylated first and then ubiquitinated. So is it the same nation that you showed us of E1, E2, E3? Okay. So it
Yeah.
just gets phosphorylated first and then it goes to that cycle.
Yep, so, okay, so I think that where we're having confusion is like where in E1, E2, E3 is this phosphorylation coming in, right? And so there's multiple things that have to happen for ubiquitination reaction. And the main thing is the ubiquitin ligase needs to be in contact with ubiquitin that's able to be transferred to the substrates. And so E1, E2 is really about activating that ubiquitin and bringing it to the ubiquitin ligase so it's on and ready to go. The phosphorylation has to do with recognition of the substrate by the ubiquitin ligase. So it's two separate things. One is the ubiquitin ligase able to polyubiquitinate, and the other one is it able to interact with the substrate. Okay? We can talk more deeply about that at some point. Okay, so this is a close-up of the figure I just showed you, right? So during G1, you have your ORC, your origin recognition complex, and it binds to your origin. So you have to imagine this is happening on all your origins, right? And CDC6 and CDT1 bind and help to load the MCX complex, MCM complex, which is your helicase. Right, you need your helicase for DNA replication, right? What does helicase do? I'm making an assumption you know what helicase is. What is helicase? What is it? Someone raise your hand. Yes.
It unzips your genes.
Unzip your DNA. Yep, don't just think about your genes. Unzip your genes. Now you'll all know. It unzips your genes. You hear that, President of Towson? Okay. This is all going to be on YouTube, but unzip your genes. Well, you know what? Whatever works, right? That's what I say. Okay. It unzips your genes and the other DNA. So important to load your helicase. So the whole purpose of this really is loading your helicase at your origins, but it's not active. And so in S phase, your SCDK gets activated, and then it actually triggers DNA replication to begin through disassembly of this complex. So that's through phosphorylation and degradation of CDC6, and then through inhibition of CDT1 by geminin, which I'm not going to go into, but that's APC actually is involved in regulating geminin. So at this phase, APC is off. Yes?
It's arranged in the first G1 phase, but why would you just undo what you just arranged?
Well, the purpose of this really is you want to load your helicase, right? And so you're getting rid of the helicase loading proteins, but your helicase is still there.
Oh, so it's
Yeah.
just for the purpose of putting it in.
Yeah, so you load your helicase and it's there and it's waiting. And then you get rid of all the other stuff that's in the way, leave your helicase, and then you can recruit your polymerase and other proteins associated with active DNA replication. So then with S phase, you get disassembly, you get recruitment of your pre-initiation complex, you get your polymerase coming, and then you phosphorylate your origin recognition complex and activate DNA replication. And then you get completion of DNA replication, and then, so now you're showing your your daughter strands of DNA. Okay, so I was thinking at one point, like, okay, so I keep telling you guys SCDK does this, SCDK does that, it's important for DNA replication, and I'm sure some of you are sitting around saying, well, what does SCDK phosphorylate, right? Because the cyclin CDK thing and activation of different phases of the cell cycle, that all makes a lot of sense, but to think a little bit more deeply, you need to think to yourself, okay, well, what is SCDK doing, right? It phosphorylates things, but what does it phosphorylate? So I looked up a few substrates of SCDK, and I thought you might find them interesting. One was CDC6, right? So I just told you about that one, right? That's that one. Also, CDK7 came up. That's a CAK, so that's cyclin activating kinase. So you can imagine that phosphorylation of CAK somehow regulates it. Does that make sense? So usually with phosphorylation, you're turning things on, you're turning things off, you're creating active sites, you're covering active sites, you're relocalizing things, all of those things. So a CAK actually gets phosphorylated by a CDK, regulation of that CAK. Here was one, FOXM1. Now you're saying to me, do I need to know all this? No. This is more just like, for me, it helps me to understand things when I can see what are some of these substrates, what are they doing, and I can put in my head how it makes sense that it's involved with progression of S phase, right? Because this is an SCDK. So FOXM1, I looked that one up. It's a transcription factor that regulates cyclin expression, right? What cyclin are you going to need next after S phase? You need M, right? So there's something that's being turned on during S phase that is then going to activate the next phase, because M cyclin starts building up during S phase. Pretty cool, huh? Histone H1, that's phosphorylated by SCDK. And so you might say, well, histone H1, what's that? And so I looked it up because I couldn't remember either. I knew it was a histone studying it. And that's the linker histone, right? And that's involved with chromatin condensation and decondensation. So when you phosphorylate, you're actually decondensing chromatin. Is that useful for activating S phase? Yes. Do you want condensed chromatin when you're trying to copy your DNA? No. Can you get any enzymes into condensed DNA? No. So that's totally logical. I saw that one and I was like, oh, that's cool. This is what I do while I eat my lunch. Okay. Here's another CDC7. MCM2 regulates the MCM complex. We just talked about MCM complex. That was the helicase that unzips your genes, right? And so MCM2 regulates the MCM complex, right? So regulates helicase. It's phosphorylated. So if you look at, let's go back to this picture. Does it show any phosphorylation or it doesn't? This is a very simplistic drawing. But you can kind of talk yourself through why phosphorylation of your helicase by SCDK might make sense. Maybe it's involved with activation of your helicase in some way, right? Your helicase is just sitting there, yeah? But you want it at some point to work and move and do something, right? What else? I saw another one, RB. We talked about RB briefly. That was the inhibitor of E2F, which is involved in activation of S phase gene expression. And it normally gets phosphorylated so that you can liberate E2F. Another one I thought was cool was Staphmin. For those of you who took 408 with me, Staphmin is that protein that binds to microtubule subunits and keeps them out of circulation. So if you phosphorylate Staphmin, then that kind of frees up all those tubulin subunits. What's going to happen next? You're going to end up going into M phase. Maybe it has something to do with activating a pool of tubulin as you prepare to enter M phase. Maybe. WEE1, that's involved in negative regulation of CDK that we learned about just the other day. So WEE1 can also be phosphorylated. And I believe I tried to figure out what the phosphorylation of WEE1 does, and it has to do with localization of WEE1. So sometimes it's nuclear, sometimes it's cytoplasmic. And so if you phosphorylate it and make it cytoplasmic, can it function? No, right? Because its substrate is nuclear. So just kind of cool things. There's so much to know, but sometimes it's just cool to kind of dive in a little bit deep to appreciate all that's out there. And that's my public service announcement for SCDK. Okay, so our major steps of DNA replication. Okay, we just went through them. We're going to get deeper and deeper here. Helicase loading, priming, sliding clamp loading, DNA polymerase is loaded. Then you get your bidirectional fork movement, right? So that's these replication forks, bidirectional, move in both directions. Sometimes people get so focused on the fact that they go one way, they forget it's actually a replication bubble with two forks. Elongation, and then DNA ligase is going to seal up gaps after the primer's removed. Okay, I'm kind of glazing over this because we're going to go into it deeply, but also you've learned this before. And in recitation this week, we are looking at this very deeply. Question?
So if it's moving in both directions, are a leading and lagging strand like relative? So can you have a leading and lagging strand on the same strand technically?
No, so you're, yes. So the question is, if your replication fork is moving in both directions, does that mean you have a leading and lagging strand on each strand of DNA? Yes, because in one direction it's your leading strand, and the other direction it's your lagging strand. Yeah. Okay, so here's where I tell you to forget everything you've ever learned before. So in 201 and 309, you learned about DNA replication in a very simplistic way. You learned about like all these different proteins that assemble, and you've got your leading strand, and you've got your lagging strand, and basically you saw something that looked like this. Your helicase opens your DNA, and then you have DNA polymerase adding bases. That's easy. And then you've got these Okazaki fragments, and your primase and DNA polymerase, and it's all happening all over the place. But what I'm going to tell you is that's not really how it happens, because your DNA polymerase functions as part of a large multi-protein complex. So in reality, your DNA polymerase and your helicase and your primase and your clamp loader, those are all kind of functioning as one complex together, moving in one direction. And I'm just warning you, you're going to have to think about this in recitation and think about it deeply, because you've seen this one so many times, you just want to think about it this way. But that's not really what's happening.
So one replication bubble has one helicase and two DNA.
Yeah, so one replication fork. So right here you're seeing one replication fork. The bubble would kind of be over, the other side would be over here. But this, I'm showing you one replication fork here. This would be the bubble up here. But one replication fork is going to have one helicase, two polymerases, two sliding clamps, one primase. But
Bottle would have two helicases and four DNA.
one bubble would have two helicases, 4 DNA polymerases. Yeah. But don't worry about the bubble just yet. Just worry about a fork for now. Yes?
What's the difference in
So, yep, so the thing is, and so we'll go through this in detail in recitation, so I'm not going to spend too much time, but I do just kind of want to have you thinking about this now, so that when you go to read your paper and go to do your recitation stuff, you'd be like, okay, I know what to do. So in this case, here's your parental DNA up here. And then so you have one DNA polymerase here, then you have one DNA polymerase here for your lagging strand, then you've got your primase here, and a helicase here, and it's kind of all over the place. But in reality, and that kind of makes no sense, so once you know that they function as one protein complex, this suddenly makes no sense, because you have everything going in different places. And in reality, what you have is your helicase bound to your different polymerases, bound to your clamp and clamp loader. And so here's your parental DNA, and your leading strand is just how it should be. But your lagging strand actually loops out and around. And that allows the whole complex to move in the direction of the fork while remaining a complex and everything being polymerized in the right direction. Yeah.
So where is the second sliding clamp part of that?
So here's one sliding clamp, and here's another sliding clamp, and here's your clamp loader, which actually binds to the sliding clamp. And this one, what happens here is this keeps coming on and off as you finish polymerization. So you have your primer, and you add to your 3' OH, and then you finish at your next primer. Then you release, and then this whole thing loops around again, and then that primer that's being put on up here kind of lines up with your polymerase, and you fills in again. Okay, and you'll have plenty of time, so don't freak out right now about it, because you'll have plenty of time on Thursday to look at it more deeply, but I'm just kind of putting it out there. For some people, it's very simple and it makes a lot of sense, and for some people, they really need to stare at it for a long time to get it, but it actually is a much more logical way for things to happen, rather than just having all these DNA polymerases running all over the place on your DNA.
And the complex is moving, or
The whole complex is moving, so it's kind of all moving with the helices. Okay? Okay. And so someone said to me, wait a minute, this is what we learned in 201 and 309, and this is what we learned in 409, and I was like, yeah. And it's just very unfortunate that I have to reprogram you, but I do. It's not that it's wrong. I think it's a good way to introduce a concept in a very simplistic way. Okay. So let's talk for a little bit about DNA polymerase itself, the enzyme itself. So Dr. Ham always starts his classes with having everyone bring in an oven mitt, because DNA polymerase looks like a right hand. So if you look at your hands, you have DNA polymerase, right? So it's kind of like So I wasn't going to steal the oven mitt idea, but it's kind of cool. So if you think about it, your palm is your active site. Down here, like at this lower palm tumor that this thing has, is where it would be the exonuclease activity. And then your incoming nucleotides come in between your thumb and your fingers. So this is a close-up of what's actually happening. So this is all happening in the palm of your hand, which is the active site. And so that's where the actual catalysis of the nucleotide addition happens. And so some things that are important is in the active site, you have these two aspartic acids that are in the DNA polymerase protein. And they coordinate these magnesium ions. And they coordinate it in such a way that it interacts with the 3' OH of the, where's my 3' OH? Of my growing strand. There's my 3, oh, see, you can't see it. So you guys are going to have to look on your own computers, because I'm missing a whole half of my DNA. There's actually supposed to be DNA here. Here's 5'. Do you guys see it on your own computers, maybe? Okay. Sorry about that. As long as you can see it. So there should be nucleotides here base pairing with these guys. But this is the one we're going to concern ourselves with. This is the new one coming in. And then this is the one before. So your 3' OH is right here. And so what happens is you have a nucleophilic attack. I'm sorry. I don't do chemistry well. But this is why you've had to learn these things. There's a nucleophilic attack of this 3' OH right here on the alpha phosphoryl group of the incoming nucleotide. And then you get that phosphodiester bond. And so that's why I always say you need the 3' OH. That's important. That's why you have the primer, because that provides a 3' OH for adding your nucleotides. That's why you always add your DNA. That's why your DNA replication happens 5' to 3'. Yeah. Questions about this? You've all seen it before. Okay, good. Okay, so another way to look at it is you've got your template strand and your primer strand. in the case of your lagging strand, or just your growing DNA molecule. And here's your 3' OH, and that OH group is going to attack your phosphate, and then you'll lose your pyrophosphate, and that's how you have your linkage between your growing DNA strand and your new nucleotide. And it all happens inside of your hand. So if I see you all going like this during the exam, I promise I won't think you wrote things on your hand. Or I will come and look at your hand. Yes?
So, what happens during nucleophilic attack?
So that has to do with that's what happens during nucleophilic attack. Doesn't that seem scary?
They break the bonds.
It has to do with taking electrons away. Who's taking orgo and wants to explain this in a really nice, deep way? Yes.
I'll say it for myself.
You're always full of fun things. Okay. No, so what it has to do with is taking the electrons. I guess these have like extra electrons, and they're taking this positive charge is pulling electrons away. So then this needs more electrons, so it makes a bond here between the O and the P. Is that good? Something like that. I'm a biologist. I always think of it as moving electrons around and grabbing things so things equal out. Is that how it works? Yeah. Okay. Good. But really, this is what's happening. Okay.
We can draw that as an answer on the exam.
You can draw that on the. If you can draw that on the exam. Sure. Oh no, everyone's going to draw that now. Okay. Okay, so this begs the question about why are nucleotides added to the growing strand 5' to 3' and not 3' to 5', and it has to do with a leaving group, and a pyrophosphate's a good leaving group, and that's what leaves when you add 5' to 3'. But if you go 3' to 5', I don't know what's going to happen here. There's like no good leaving group. I guess you can say like this would want to attack. I don't know what would happen. So that's why it doesn't happen that way. Okay. Yes.
So I was completely confused by the previous slide.
That one?
No,
The red one is angry.
I just thought of the bad leading group versus the good leading group.
That is because you
So
want
it
to,
exposes the OH group, right?
huh?
Like you have 5' to 3', it exposes the OH group.
Well, so the OH is going to attack, the oxygen basically is going to attack the phosphate, and then it pulls the electrons away, and that makes the other the pyrophosphate, leave. That's the leaving group. And so that's a good leaving group. But here, you've got your oxygen, if it attacks this phosphate, which is what I guess should be happening, there's no good thing it's not a good leaving group. So it's not a favored reaction. I should have a chemistry professor come down for this part. That's my understanding, and it makes sense to me.
Basically, in the good living group, as far as I'm seeing, when actually the good living group would be water, H2O. In the end, we have H2O leaving.
Over here?
Yes. No, over.
So here you have the pyrophosphate leaving. The two phosphates are leaving. Okay. Okay. How are we feeling about leaving groups? Okay. So DNA is synthesized 5' to 3' because A, that's what I was told in 201. It's true. No. B, the 3' OH on the most recently added nucleotide is available to attack the alpha phosphoryl group on the incoming nucleotide triphosphate. Or the 5' OH on the most recently added nucleotide is available. And so it's a 3' OH that's available to attack the alpha phosphoryl group. And D is, I know sometimes you feel that way, but and that kind of speaks directly to this right here. That 3' OH right here is going to attack that phosphate group, and the pyrophosphate is going to be. Questions? Bless you. Everyone's speaking today. Okay. Let's talk about fidelity. So and we're just talking about fidelity of DNA replication. We don't need to talk about any other fidelity. I was thinking of my retirement fund. I don't know what you guys were thinking about. That's scary, by the way. Is when you have a retirement savings. Because that means one day you're going to retire. Which is mind-blowing. Anyways, because, yeah. So the fidelity of DNA replication is important. The fidelity, when I talk about fidelity, I mean that is it correct? Is it error-free, or are there a lot of errors? Something that has high fidelity, not talking about record players, something that has high fidelity is going to copy things properly, putting in the right base, and not make mistakes. Something that has lower fidelity is going to make a lot of mistakes. And so that's why when we talk about fidelity of polymerases, we talk about it in terms of the error rate. Something with a very high error rate is going to be have lower fidelity, right? So the error rate for replicative polymerase, so that's the DNA polymerase that replicates our genome, is about 1 in 100,000. That's pretty good. That's one error in every 100,000 bases that are polymerized. And so fidelity is maintained at 2 different stages. One is during polymerization, and that has to do with the space in the active site. The right base fits better than the wrong base. And then after polymerization, because of the proofreading capability of DNA polymerase. And I think I'm going to stop here because I'm seeing people packing up and putting jackets on, and it's about only 2 minutes left. Okay.